So good morning. For those of you who don't know me from previous years, my name is Andria, and I'm a postdoc at the University of Lausanne. For this year's talk, I'm going to take you to some brand new results where we're looking into whether auditory stimuli synchronized to the ongoing heartbeat can actually modulate background slow oscillatory activity in sleep. So in our lab, we actually generally study how the human brain processes sounds across a variety of consciousness states. And what we have shown over the years in a series of EEG studies is that indeed the human brain takes advantage of the temporal cue provided by the ongoing heartbeat rhythm to help it identify patterns among such external auditory stimuli in wakefulness, sleep, and coma. Marcia will take you over these results thoroughly tomorrow, so for today's talk, I'm just going to focus on sleep, which is unique among these consciousness states in that it's characterized by rich changes in background oscillatory activity, particularly in the deepest stages of non-rapid eye movement sleep. One example of such oscillations are the so-called slow oscillations, which are periods of global synchronous neural activity oscillating between 0.5 and 1.2 hertz, observed in the deepest stages of sleep N2 and N3. These are characterized by peaks of increased neural excitability and troughs of neural acquiescence. Now, why are they interesting? Because they've been repeatedly implicated in information processing during sleep and of relevance to us in implicit learning during sleep. Now, specifically in the context of cardiac and auditory stimulus processing, we looked into two lines of investigation related to slow oscillations. The first comes from the closed-loop auditory stimulation literature in sleep, where, as you can see, sounds are administered at the peaks of the slow oscillations in real time, which results in induced trains of slow oscillations that follow the auditory stimulation. The second line of evidence comes from the investigation of the coupling between the brain and the body in sleep. This seminal study, looking at the distribution of our peaks along the slow oscillation cycle, shows that the peaks, our peaks tend to cluster around the peaks of the slow oscillations, suggesting that indeed there is some form of coupling between the brain and at least the cardiac signal, as you can see here in this case, even in deep sleep. With this in mind, here we wondered whether if we actually experimentally imposed a fixed interval between the R peaks and the sounds, can we observe similar modulation of slow oscillations in deep sleep? To do this, we utilized a commonly employed paradigm in our lab where cardiac and auditory stimuli are either synchronized or not. In the appropriately termed synchronous condition, we impose a fixed interval between the R peaks in the ECG signal, which we detect in real time, and the sounds that we administer. We also administer a purely external auditory regularity condition with fixed sound-to-sound intervals based on our participants' average heartbeat rhythm. Our control irregular condition comes by replaying what we generated in a previous synchronous block, now having no direct relationship to the current ongoing heartbeat, and therefore resulting in variable R peak to sound and sound-to-sound intervals. Now, for the study I'm going to show you today, we administered these trains of auditory sequences along with the baseline condition without any auditory stimulation in a cohort of healthy volunteers while recording continuous electroencephalography and electrocardiography. We recorded 2 consecutive nights of sleep, and for the purposes of this study, extracted the periods of N2 and N3 sleep and concatenated them. So after basic preprocessing, we attempted to isolate the slow oscillations in our signal using standard criteria. This gave us the average slow oscillation waveform, having the characteristic frontal polarity. And for this reason, we chose for this study to focus only on the frontal electrodes. So any results that you see will be the average over all these frontal electrodes that we focused on. The last step before we get to the results was to actually identify which experimental condition each of the slow oscillations belong to. Once we had this, we could ask the main question here. Do slow oscillations actually reorganize as a result of the auditory stimulation in deep sleep? First and foremost, in the basic features that characterize slow oscillations, that is their amplitude and their duration. So for the peak-to-peak amplitude first, just to briefly explain the figure, as there's many of them coming up in upcoming slides, what you're seeing is the single subject median amplitude in this case, along with the, no, wrong button, along with the average values and the results on the right-hand side of the post-hoc statistical analysis comparing our experimental conditions. So, right button now. What you can see is that, but it's, I want to see the crowd. Allora, so, as you can see, there you go. Ah, it's true, I was pointing there. I just got it. Okay, so, IQ of a potato. So, what you can see is that in the conditions of auditory regularity, we have higher amplitude compared to the control conditions where no such regularity exists. More interesting to me is actually the duration, where you can see that across all of our auditory conditions, we show a reduced duration for the slow oscillations compared to the baseline condition without auditory stimulation. Now, this is interesting if we think about the experimental paradigm in this study, in that the sound-to-sound intervals actually resemble the heartbeat rhythm, which in sleep is about 1 second, suggesting that if there is indeed some alignment of the sounds in response to the auditory stimulation to the slow oscillations, they could actually become shorter to align with the sounds-to-sound interval. Now, with this in mind, we could actually go on to ask an even more interesting question. Is the temporal relationship between the sounds and the slow oscillations actually modulated by the auditory stimulation? To do so, we identified the temporal interval between the peaks of the slow oscillations and the administered sounds. What you're seeing here is indeed the slow oscillation waveform for our 3 auditory conditions, now time-locked to the sound onset. And we investigated essentially the latency between the peaks of the slow oscillations and the administered sounds. What we saw is kind of interesting again, in that this, as you can see, the latency tends towards 0 in the condition where cardiac and auditory stimuli are synchronized, suggesting perhaps that the brain may be using these periods of maximal excitability to process the auditory sequences in this highly relevant to it cardio-autosynchronous condition. Now, we cannot exclude that it's simply coming from the experimental manipulation in this study, based on the fixed interval that we impose between our peaks and sounds. To make sure that this wasn't the case, we performed exactly the same analysis, now looking at the interval between the peaks of the slow oscillations and the R peaks in the ECG. And here, I want to point out 2 interesting results, at least to me. The first is that, as you can see, the synchronous condition is different to the remaining auditory conditions and the baseline condition, suggesting that whatever we just saw here for the auditory comparison is possibly driven by the sounds indeed. The second interesting observation relates to the remaining auditory conditions and the baseline, where we see that indeed the latency between the peaks of the slow oscillations and the R peaks tends towards 0, resembling essentially the results that I showed you at the start of this coupling of the R peaks to the peaks of the slow oscillations. Now, with this addressed, in our paradigm, we had the unique opportunity to actually see what happens over the 2 consecutive nights of administration of these sounds in our participants. Does the repetitive stimulation actually modulate differently the slow oscillations? Exactly the same analysis, now separating the slow oscillations in the 2 nights. And what you can see is that while on the first night we have a similar observation to what you just saw when we put everything together, in that the synchronous condition tended to, the latency rather, tended to align with 0. This was not the case on the second experimental night. For this, I'm just speculating here, it could relate to some degree of adaptation, of habituation to the auditory sequences, and perhaps the brain no longer needs to rely on these periods of maximal excitability to process these types of sounds. Now, just to put all these soup of results together, what it does show, at least to me, is that indeed the slow oscillations reorganize as a result of the auditory stimulation in this study, specifically when the auditory stimuli are locked to the ongoing cardiac rhythm. This resembles our earlier findings, where we saw similar tendency for the cardio-autosynchronous sequences towards 0 in the same latency, this being done only in a sleep in a smaller cohort of volunteers and for 1 acquisition night only. I think the general point here is that indeed the coupling between the brain and the body is of relevance beyond wakefulness into states of altered consciousness. And I think is similar to Diego's point, actually, in that we can use these sort of background oscillations, these types of metrics, to make observations about covert consciousness in the absence of behavioral responsiveness. Voilà, that's it. Thank you to everyone involved in the study. Thank you for listening. No more peak words.
Thank you so much, Andria. So do we have any questions from the audience? Yeah, Leah. Oh, it's you. Ready?
Hello. Hi, that was an awesome talk as well. I was just wondering, I'm not sure, I missed maybe what your control trials were, because I couldn't help but think between the synchronous trials with the heart and the regular, could some of the results be driven to some extent by the variability in the heart-related condition versus the regular? But maybe this is accounted for in your control, because I couldn't remember what the control one was.
The variability in the sound intervals?
Yes.
So what we do in the control condition, we replay exactly the same sequence that we generate in the sync shuffling. So the variability across the trials is the same. Actually, it's similar in the regular condition. You have less variability between sounds, but the overall distribution, let's say, of the intervals is equal. So somehow you can control for this to some extent. Yeah, but you cannot exclude it. Huh?
That was exactly my question, thank you.
Okay, then there's a question here in front.
Very much. I was wondering if you were thinking about what that functionally means. I mean, you started your talk with talking about the role of sleep and plasticity in learning, in regulation of homeostasis maybe. Have you any tests on behavior in that study?
In this study, we also did some sort of a behavioral examination in that after the sequences, for one of the nights, we also played some instrument sound. You can hear me? Okay. So we played some instrument sounds, which we then tested on the upcoming morning in terms of whether they could be recalled better than sequences that were not played during sleep. One of the next steps here is to actually look into the relationship between the slow oscillations and the played sounds to see if it's perhaps driving the accuracy and the performance of our participants the next morning.
Thank you. Yeah, thank you. So I have two short questions. One is, I understood you have a look only at one electrode, but was there any topography? So maybe the strongest effect was somewhere else. I know that maybe the slow waves are there where you look, but the interaction could be strong elsewhere. And the second point is that I noticed tremendous variability across subjects.
Yeah.
So did you observe that your strongest effects were for those subjects who had stronger slow wave, or was it dependent on it or not?
So this is the next steps, actually, to look at the correlation between how the capacity, let's say, of our participants to have these high amplitude slow oscillations and to what extent, and compare them to their relationship, the brain-body coupling, if we want to call it that. So for your first question, yes, we performed the analysis over all the electrodes. What I'm currently doing is essentially running cluster permutation statistical analysis of the same kind of waveforms to look at whether there are clusters where this activity may be localized that extend beyond the frontal electrodes, as I did first here.